Okay, so lab 5 was more using the microscope and being able to observe different microorganisms and cells under the magnification. So one of the big things to remember was that, let's see if I can draw a microscope well, I don't know. So say that's like the eyepiece, it's all this busy stuff, and then you have those rotating lenses and the whole thing. Yeah, not an artist. Remember that these are at 10x, one here is at 4x, one here is at 10x too, and one here is the highest magnification, 40x. So the way these microscopes work and the lenses work is that this 10x is magnified by one of these, whichever one you've switched it to. So if you multiply 10 times 4, 10 times 10, 10 times 40, you get the magnification that you're looking at here. And we were able to use that measuring slide to see the actual width in micrometers, I believe, to kind of observe what we were going to see at what size. So in the cases of, here, let's get rid of this. So prokaryotes, eukarya, so that's short for eukaryotes, but let's write eukaryotes. Large, these were much larger than their cousins. Complex, and by what I mean by that is that they have organelles, nuclei, none. So these are not complex. DNA, it's in the nucleus. DNA in prokaryotes and bacteria, it's just free. It's just free floating. So these are some of the main concepts. Prokaryotes are unicellular. These are multicellular, and that's more general. Eukaryotes can be single. Prokaryotes can kind of collect sometimes, but these are your general rules. What we observed mainly, though, was the size, because we saw that at the 400x is what we had to use. 400x was to use for prokaryotes, and we could usually get away with 100x or even lower looking at the eukaryotes, so we could even do those that 40x. Okay, and some of the main features that we were able to see were essentially cell size, the shape, and the lack of a nucleus. So animals versus plant cells. Remember how these were stacked on each other? They had their nucleus, and sometimes you saw a big vacuole. That's where plants can store a lot of their H2O, and that's why plants are rigid, is they can stretch these vacuoles. And one of the other keys is that plants have a solid cell wall, and they have a plasma membrane. And shoot, I don't have a green marker, but plants also have chloroplasts, so imagine this blue is green. Now, both of them have mitochondria, but animal cells do not have any chloroplasts. Chloroplasts essentially take CO2, they convert it into chemical energy like glucose, or a glucose, like a type of, like a substrate that mimics glucose, and then that glucose still goes into the mitochondria and does the same reaction that ours does when we consume glucose. Yeah. So the other big thing for this one was tonicity, and that's an osmosis, and that's when, so say this is a potato cell like we saw. If there's just regular H2O out here and H2O inside the potato actually has a pretty high salt concentration inside it. So the infinite amount of water that's outside that was in your tray, and you can kind of imagine it as infinite. So this is our cell. That water will go inside to meet this solute. Essentially, it's trying to down concentrate this solute by making it bigger, or by adding more water, the concentration of salt in here is going to be lower inside the potatoes. Say though that although there's a little salt in here in the potato, if we add a solution that has lots of NaCl, tons, what we saw was our potatoes actually shed their water. Their water went out to meet this solute. It followed the solute to concentrate it down to the levels that it's normally at right here. But the salt in here, it can't cross back and forth. So it was stuck, and we saw our potato cells lose their water to meet this large amount of salt. It's a little. So there's tons of salt out here in that solution. H2O went out to meet it, and our potato cells kind of shriveled.